Your focus now should become like a cold-blooded murderer. Intense, but not jumpy. Select the wrong statement. Isogametes are similar in structure, function, and behavior. As we look at it, analyze it. Iso, similar. Anisogametes differ either in structure, function, or behavior. In Oomycetes, the female gamete is smaller and motile, while the male gamete is larger and non-motile. Chlamydomonas exhibit both isogamy and anisogamy, and Fucus shows oogamy. Now, there is a possibility that you might not know Chlamydomonas being isogamous and anisogamous both, and Fucus showing oogamy. What can you be certainly sure is oogamy is where female gamete is larger and male gamete is smaller. Statement number 3 says that in Oomycetes the female gamete is smaller and motile while the male gamete is larger and non-motile. That is wrong. The female gamete is larger and non-motile, male gamete is smaller and motile. But the statement is opposite. It is stating the opposite. Iso and anisogamy. Isogamy is when both the gametes are similar. Both the gametes are similar. Anisogamy is both the gametes are different. Alright. Which one of the following is not a correct statement? Herbarium houses dried, pressed and preserved plant specimens. Not a correct statement. Is it correct? Herbarium houses dried, pressed and preserved plant specimens. Herbarium is a dried storage place, dried specimen storage space of plants. Okay, that statement 1 is correct. Botanical gardens have a collection of living plants for reference. Is it correct? Correct. Where what kind of plants do we cultivate in botanical gardens? Plants of significance and plants which are endangered. Plants which cannot probably survive in the wild are protected and preserved in the botanical gardens. A museum has a collection of photographs of plants and animals. True? Can there be photographs of plants and animals in a museum? Or should not there be photographs? Let us see the next one. Key is a taxonomical aid for the identification of specimens. Key is correct. What is key? It is like your algorithm. Keys are like algorithm. There is a key. Is it a plant specimen? Yes or no? If it is yes, then there is another key. Key is basically a question or an indication. A question or an indication, a statement to which there are two responses. Yes or no? Key either opens the lock or does not open the lock. That's all, no? So there are two options always for a key. Algorithms are also like that. Algorithms are also like that. You have a statement. There are two opposing options to choose after the statement. Is the person a male or a female? Male. Less than 18 years or more than 18 years? Less than 18 years. In school or not in school? Not in school. So goes on like that. It's a demographic survey. So the person is out of school. Alright. Literate or illiterate? Literate. That means what? A person who is less than 18 years out of school currently but is illiterate. Indicates that the person has gone through something or something has happened which has made the person get out of school. It gives you some indication. Likewise, key is also like an algorithm which will help you put organisms that you identify into a particular category. So it is correct. Key is a taxonomic aid for the identification of specimens. But museum has a collection of photographs of plants and animals. Is it wrong? Is it a wrong statement? Can there be photographs in a museum? There can be photographs in a museum. So what is wrong in the statement? The right choice is statement 3. The right choice is option 3, which is a wrong statement in this context. So here we have to read it as museum has only collections of photographs of plants and animals, which is not right. There will be specimens also in a museum. Museum also houses specimens. All the other three statements are correct, absolutely. So in that sense, an incompletely correct statement can be considered as a wrong statement. Alright, looked at by itself, this is not a wrong statement. Museum has a collection of photographs of animals and plants, that is also true. But not only photographs, it will have specimens also. Isogamous condition with non-flagellated gametes is found in. Now these are examples. Have you made note of examples from NCERT? Isogamous with flagellated gametes, isogamous with non-flagellated gametes. Difficult for me to remember this kind of stuff. Alright, but with respect to the questions, I can remember. But a list of things as examples, difficult for me. So strategically, I leave out things that are dependent only on memory. I leave out things because I always look at effort and outcome, risk-benefit analysis. The risk or the effort that goes into remembering line items for which I don't have a natural tendency to remember. I can't remember names and dates. So I will have to put in a lot of effort to remember names and dates, which I choose not to. Examples given in NCERT, remember. Examples given in NCERT, you remember. Alright. So isogamy is what? Isogamy is similar gametes, morphologically similar. Spirogyra, isogamous reproduction with non-flagellated gametes. Spirogyra, isogamous with non-flagellated. Others are Chlamydomonas, gametes are flagellated and motile. Volvox and Fucus, anisogamy or oogamy. Alright, isogamous and with non-flagellated is Spirogyra here. You will have to remember the examples from NCERT by repeated revision. I don't have any shortcut to remembering names, examples. Besides paddy fields, cyanobacteria are also found inside vegetative part of Pinus, Cycas, Equisetum, Celotum. Where else do you find cyanobacteria? What is cyanobacteria? Why is cyanobacteria significant? What is the significance of cyanobacteria? They are nitrogen fixers. Cyanobacteria can fix nitrogen. So cyanobacteria, nitrogen fixing bacteria establish what kind of a relationship with other organisms? Plants primarily. Symbiotic relationship. Okay. What specific cyanobacteria do you study as examples for nitrogen fixation? Anabaena. Alright. Anabaena, nostoc. These are examples given in your NCERT book. Anabaena and nostoc, you remember these two examples for cyanobacterium. With which plant do they establish symbiotic relationship here? Cycas. The root of cycas is special. What kind of root it is? Coralloid roots. Coralloid roots. There are nodules. Coralloid roots is found in cycas. Cycas has coralloid roots. In coralloid roots, there will be nitrogen fixing cyanobacteria present in a symbiotic relationship. Alright? Equisetum is what? Horsetail. What is it? Pteridophyte. Okay. Megasporangium is equivalent to embryo sac, fruit, nucellus, ovule. Megasporangium produces what? Megasporangium produces megaspores. Megaspores are like seeds. Sporangium produces megaspores. Megaspores are like seeds. Now those are the reproductive propagules. Megaspores are the reproductive propagules. In angiosperms, what is the reproductive propagule? Seed. If megaspores are equivalent to seeds, seeds develop inside what? Seeds are inside what? Ovule. Seeds are inside the ovule. Ovule is inside the ovary. Ovules are inside the ovary. So ovary is equivalent to the fruit. Ovule is equivalent to the ovary is the entire fruit. Ovule is again the equivalent to the megasporangium, which will produce multiple spores. Each ovule develops into a see, ovule is the one which will become the seed, no? Ovule becomes the seed, ovary is the fruit. Ovule becomes the seed, ovary is the fruit. Embryo sac is the one which will house multiple ovules. Embryo sac holds multiple ovules. So megasporangium is found in what? Megasporangium is found in gymnosperms. Megasporangium is found in gymnosperms. Megasporangium is the one which will produce megaspores, which are the seeds. So shouldn't we consider megasporangium equivalent to the embryo sac? No, each megasporangium itself is the seed. Megasporangium itself is the one which develops into a seed, so equivalent to the ovule. Megasporangium is equivalent to the ovule. There is nothing equivalent to the ovary as such in the gymnosperms because there is no fruit development. Read the following statements A to E and answer the question which follows them. In liverworts, mosses and ferns, gametophytes are free living. What type of life cycle do you find in liverworts, mosses and ferns? Haplodiploidic. Haplodiploidic means what is dependent, what is independent. What is dependent, what is independent? Gametophyte is independent, sporophyte is dependent. Gametophyte is independent, sporophyte is dependent. So whatever is independent is free living, whatever is dependent is depending on the free living form. So in liverworts, mosses and ferns, gametophytes are free living. Liverworts, mosses and ferns are what division do they belong to in the plant kingdom? Bryophyta. In bryophytes, it is free gametophyte and dependent sporophyte. So statement number 1, statement A, is it true or false? True. Gymnosperms and some ferns are heterosporous. Heterosporous means what? Heterosporous means two different types of gamete spores. Gymnosperms, yes, megaspore and microspore is produced. Some ferns are what? Pteridophytes. Pteridophyte. We have seen that pteridophytes exhibit the precursor to seed habit. Pteridophytes exhibit the precursor to seed habit. So in some ferns also we can see the heterosporous nature. That statement 2 is also true. Statement B is also true. Sexual reproduction in Fucus, Volvox and Albigo is oogamous. We have seen earlier also in an example that Volvox, Fucus and all have oogamy. So sexual reproduction is oogamous, that is also true. A, B, C are true. The sporophyte in liverworts is more elaborate than that in mosses. False. Elaborate. Both Pinus and Marchantia are dioecious. What are dioecious plants? What is dioecious plants? Dioecious plants produce what kind of flowers if they produce? Unisexual. So dioecious plants are unisexual or bisexual? Dioecious plants are unisexual or bisexual? Unisexual. Monoecious condition is bisexual. Monoecious condition is bisexual. Pinus is monoecious. Pinus, pine trees are monoecious. Marchantia is dioecious. Separate male and female plants. So statement E partially correct. So it is false. How many statements are correct? 3 statements are correct. Oh, this is again a question. Among bitter gourd, mustard, brinjal, pumpkin, china rose, lupin, cucumber, sunhemp, gram, guava, bean, chili, plum, petunia, tomato, rose, withania, potato, onion, aloe and tulip, how many plants have hypogynous flowers? Huh? +4 or leave it 0. Alright. In all this, what we have to recall is what is hypogyny. What is hypogyny? Superior ovary. Hypogyny, the other words are hypo to gyne, below the ovary. Hypogyny means below ovary. What is below ovary? Ovary is not below the others. Everybody else is below the ovary. Hypogyny. Hypogynous condition is superior ovary. What is inferior ovary? What is the condition in which there is inferior ovary? So other words are above ovary. What is it? Epigyny. Epigyny. What is the condition in which all of them are in the same level, at the same level? Perigyny. That's all we have to remember. Apparently 15 of them are hypogynous. All right. Not possible to remember. No, literally not possible to remember. That means you have to know, you should have studied all the classes of these plants. Beyond the scope of your learning here. You are not preparing to become botanists. All right. So you need to know what is the different states, that's all. Interfascicular cambium develops from the cells of medullary rays, xylem parenchyma, endodermis and pericycle. See, there are multiple concepts here. Cambium, interfascicular cambium, where that means fascicle. What is fascicle? Medullary rays, xylem parenchyma, endodermis, pericycle. What is cambium? Cambium is the persistent meristematic tissue in the stems and roots, which will contribute to the development of secondary tissue, which will contribute to secondary growth. Cambium is what which contributes to secondary growth. There is primary cambium and secondary cambium. Cambium is persistent meristematic tissue. What is the role of cambium? Contributes to growth. Contributes to growth, primary growth as well as secondary growth. Cambium is what gives rise to growth. Fascicle means what? Bundle. Fascicle is bundle. So there is phloem on the inside, xylem on the outside. Xylem and phloem is there. That is one bundle. If the cambium is between the xylem and phloem within the same bundle, what do you call that? Inter and intra. Two terms you remember. Inter means between. Intra means within. Fascicle means bundle. If it is within the bundle between xylem and phloem, it is intrafascicular cambium. So there is one bundle here and one bundle here, and it goes on like that. One bundle here, one bundle here. It is all around the trunk. In the stem, all around the trunk, there are multiple bundles of xylem and phloem. There is cambium in between all of this. Unless the cambium becomes a ring, it will not lead to proper growth. So it has to form a ring of cambium. Between the bundle, within the bundle, there is cambium in between the two. Adjacent bundles, between this bundle and this bundle, there are parenchyma cells in between, correct? There are parenchyma cells in between the bundles. Those parenchyma cells between the bundles of xylem and phloem, vascular bundles, is called what? Medullary rays. Medullary rays are those parenchyma cells in between the vascular bundles. Now adjacent to the cambium, let us say this is the cambium line, this is the line of cambium, this is the line of cambium here. There is a gap here in between. Now here to here, the cells that is like the bridge between the two cambium should transform to become meristematic again, so that it completes the ring. Now between from here to here there is a gap. You have to complete the cambium ring there. Transformation of the parenchyma cells in the medullary race to form a connection between the two interfascicular cambium, forming that bridge. That bridge between the two interfascicular cambium is the intrafascicular cambium is the interfascicular cambium between the bundles. That will complete the ring. And when those cells start multiplying, they will add cells both inside as well as outside of the ring, adding to the girth of the plant body. Understand? So, interfascicular cambium, intrafascicular cambium done, medullary race we understood. Xylem parenchyma means what? Xylem has 4 elements. What are the 4 elements? Within xylem? Tracheids, trachea, xylem parenchyma and xylem fibers. Tracheids, trachea, xylem parenchyma and xylem fibers. Tracheids are independent cells, conducting cells. Are tracheids living cells? Tracheids are dead cells. If you talk about the type of cell tracheids are, what type of cells are they? There are 3 types of cells in plants that you will find based on their nature. How are they structurally? Parenchymatous cells, cholenchymatous cells and sclerenchymatous cells. Which of these 3 is most conducive for activity? Very active. Which of these 3 are very active? Is very active. Parenchyma is very active. Very flexible. It does not provide strength, but it gives a lot of functionality. Thin walls, thin membranes, thin walls, great exchange capability. It can become a reservoir of storage material. It can store food, it can store water, it can give up food and water, it can participate in exchange, metabolically very active, parenchyma. So wherever you want activity in the plant body, you will have parenchyma cells. Wherever you want to store. Dynamic storage. See, static storage and dynamic storage. Static storage means you store and leave it. You don't use it again. You are not constantly using and storing. Dynamic storage is you are storing when there is excess, and when there is a demand, you use that also. Parenchyma is very favorable for dynamic storage. Cholenchyma provides function, but because of its thick walls also, it will provide stability as well. Not as active as parenchyma cells, but not dead as sclerenchyma cells. Sclerenchyma cells are dead because of what? Because of the deposition of secondary cell wall. Secondary cell wall is made up of what? Lignin. Lignin is impermeable, absolutely impermeable. Lignin will not allow even gas exchange to happen. So if you deposit lignin around a cell, what will happen to exchange of material with the between the cell and its outside? Can there be exchange of material across the wall? Not possible. So when there is no exchange of material, there is no survival of the cell. So sclerenchymatous cells eventually die. Are sclerenchymatous cells produced or sclerenchymatous cells happen? Every cell when they are produced are parenchymatous only, because they are coming from meristematic cells. Every cell in the plant body is produced by meristematic cells. Meristematic cells become parenchymatic cells when they stop multiplying and start functioning. Then gradually there is deposition of more and more wall material. They can become sclerenchymatous also. So with xylem, 4 elements. Tracheids, independent cells. Trachea, join these cells to form tube. Xylem parenchyma for dynamic exchange. Xylem fibers for strength. 3 dead elements and 1 living element. Phloem, tell me the phloem elements. 4 again. What are the sieve tubes, sieve cells, sieve tubes, then phloem parenchyma and phloem fibers. Phloem fibers. Companion cells, are they part of the phloem elements? Do you want to consider companion cells as a part of one of the 4 elements? Sieve tube, sieve cells, phloem parenchyma and phloem fibers. These are the 4 elements. Sieve tube and sieve cells. See, it's like sieve cells are like tracheids. The difference is tracheids are dead, sclerenchymatous. Sieve cells are not dead. Sieve cells are not dead. They are living cells. If a cell is living, the forces of metabolism will act on it. So it can regulate movement. But a sieve tube, the trachea cannot regulate movement. The movement is dependent on the external forces. Alright, the gradient will determine what direction the water will flow, and the gradient is always root to leaf. That is the gradient, one directional movement. Sieve cells, sieve tube. Sieve tube is stack up these cells one above the other. Perforated walls is sieve cell and sieve tubes. A cell with perforations, perforated walls. This room, you punch holes into the room, perforated room. If this room is if this room has all the walls around it being perforated, that is a sieve cell. You stack up, line up multiple cells one above the other, multi-storied building with all the walls, all the roof and floor being perforated. That is the sieve tube. They are living. Nucleus is pushed to the periphery. Nucleus is pushed to the periphery. Why do you need companion cells along with this? Why do you need companion cells along with this?
Because sieve tubes don't have nucleus.
Sieve tubes don't have nucleus. Okay. So
Function of nucleus is regulated by companion cells.
companion cells will determine the direction of flow of material. Food being transported or the sap being transported in the phloem elements have to be transported in multiple directions. It is not one directional flow, so that you can apply the osmotic forces for bringing about the movement. It is multidirectional movement. Depending on the conditions, movement should happen. So who will analyze the condition? Some living cell should analyze what the conditions are. Based on the analysis of the living cell, the direction of movement will be determined. That is why phloem needs to have more living components. There are 3 living elements in phloem and 1 dead element. Dead element is phloem fibers. The living elements are sieve tube, sieve cells, phloem parenchyma. Why do we need parenchyma in both xylem and phloem? Local storage. As material is being transported, you are keeping this material in storage at every level. See, water is flowing from the root to the leaf. The osmotic forces, transpirational pull. There are 2 forces. There are 2 forces which is bringing about movement of water in the xylem. What are the 2 forces? One is transpirational pull theory, which explains that. One force is transpirational pull, and the other one is root pressure theory. Root pressure theory. Root pressure is creating a upward push. Transpiration is causing a pull. It is a push and pull. Push by the root pressure, pull by the transpiration. So if you just let that, all the water is just flowing from the root to the leaf and gone. In between also you have a lot of plant tissue, no? They also need to be supplied with water for their metabolism. Parenchyma stores it. It acts as a storage which will prevent loss of water and provides supply at every level. Food also the same way. Alright. So in this intrafascicular cambium, medullaris, xylem parenchyma, endodermis, pericycle. Endodermis is found in Which part of the plant body? Endodermis, pericycle, where do you find it? Is pericycle found in all plants? Pericycle, endodermis. Endodermis is the boundary between the cortex and the medulla. Boundary between the cortex and the medulla. Why is pericycle significant? Pericycle is present in shoot also. That means pericycle is present in root. Pericycle is present in root. Do we find pericycle in shoot also? Pericycle is a root character. Why is pericycle significant? Can anybody tell me? Why is pericycle significant?
So because the younger roots they come out from pericycle.
Root? What roots?
In nature roots, like for
The branching of the roots is happening from the pericycle. So if you are given a wood with a branch, how do you find out whether it is shoot or root? The lateral branch of a root comes from the pericycle, it goes deep inside. The lateral branch of a shoot is coming from the epidermis, from the surface. From the what? From the bud. It is coming from the bud. Lateral branch of root comes from pericycle. Pericycle is significant for that and it is a characteristic feature of root. Okay. All the things to be discussed in that question done. In China rose, the flowers are actinomorphic, hypogynous and twisted estivation, actinomorphic, epigynous and with valvate estivation, zygomorphic, hypogynous with imbricate estivation, zygomorphic, epigynous with twisted estivation. China rose we have to study. Actinomorphic means what? Actinomorphic is? Actinomorphic and zygomorphic refers to what? Actinomorphic and zygomorphic refers to symmetry. Actinomorphic is what symmetry? Radial symmetry. Zygomorphic is bilateral symmetry. China rose is hibiscus. Correct? China rose is hibiscus. So good thing that we all of us have seen hibiscus. What can you eliminate even if you don't remember by common sense logic? What can you eliminate in this? At least symmetry you can guess, no? By common sense logic. See, while answering question, try to figure out some things by common sense also, by your past experience, what you have already seen. Have you seen a movie by the name Slumdog Millionaire? No? Slumdog Millionaire was India's Oscar entry. Very acclaimed movie. Naturally, Oscar is a Western award. Those fellows will give award to anything that will show India in very bad light. You show poverty, you show people not living in good conditions. They say, oh, beautiful movie. Showing reality. What nonsense. The reality is other things also. Anyways, in that, the movie is like this. A slum boy, a boy who comes from a slum who is not educated and he wins the Who Wants to Be a Millionaire competition. Now there is a big issue that happens telling that how can this fellow who comes from a slum who is not educated win this competition? He wins $1 million. Like Kaun Banega Crorepati? He becomes Crorepati. An inquiry is established and the police starts investigating. For every question, how did you know answer to this question kind of thing. For every question that was asked, there is some experience in his life which relates to that question. Goes like that. Actinomorphic is radial symmetry. Hypogynous is all other worlds are below the ovary. In hibiscus, that is what happens. We see that we see the bulge, the ovary bulge above all these whorls. Okay. Twisted estivation is overlapping estivation. Twisted estivation is also called Overlapping estivation. One overlaps the other. One end is overlapping the other. One in, one out. One in, one out is twisted estivation. Alright. So radial symmetry, hypogyny, twisted estivation. Alright. Lenticels are involved in. Lenticels are found where? If the question, if I give you a question as lenticels are present in option A leaves, option B stem, option C root, option D flowers. Stem. What kind of stems? Stems which have secondary growth. Lenticels are not found in stems without secondary growth. So why are these lenticels there? To allow for gas exchange. Allow for gas exchange. The cells in the lenticels are living or dead? The cells lining the lenticels, they are living cells. So lenticels are there for the purpose of gas exchange. Lenticels are involved in transpiration, gaseous exchange, food transport, photosynthesis. They are found in the bark of woody plants. Woody plants are those which have secondary growth. All right. Age of a tree can be estimated by, these are all simple questions. Height and girth, are they dependent on the season, or height and girth is dependent on the conditions? Height and girth of the plant. Biomass also depends on the conditions, availability of nutrients, water, sunlight, all of that. Number of annual rings, diameter of the heartwood. See, heartwood and sapwood. Heartwood and sapwood are an indication of the changing seasons in which the plant has grown. Heartwood and sapwood, or hardwood and softwood, both are part of the annual rings. Both are part of the annual rings. Annual rings might be thicker or thinner, but annual rings are a clear indication of age of the plant. Rest all are even the thickness of the annual ring, the size of the annual ring, all of that is dependent on season only, season and conditions. But a more accurate representation of the age of the plant are the annual rings. Seed coat is not thin and membranous in. This is important. Seed coat is not thin and membranous in maize, coconut, groundnut and gram. Now when you look at all four, maize, coconut, groundnut and gram, which of these are monocotyledonous and which of these are dicotyledonous? Tell me. Maize and coconut are monocots, groundnut and gram are dicot. Thick seed coat is characteristic of monocots or dicots? Leguminous plants or the leguminous seed producing plants are covered by thick seed coats. That is a characteristic feature of legumes. So gram has thick seed coat. What about groundnut? Does it have a thick seed coat? Very thin paper-like seed coat. Coconut and maize. What is the seed coat in coconut? Is the shell the seed coat? Is shell the seed coat of coconut? What is that? Endocarp. Shell is the endocarp of coconut. Pericarp, epicarp and endocarp. Those are all very fibrous. That brown color coating you find on the outside of the coconut when you take it out. That is the seed coat there. Thin there. Alright. In maize and coconut, the seed coat is thin and it is fused with the pericarp. It is fused with the outer coverings of the seed. Groundnut is papery. Gram has thick seed coat. Transition state structure of the substrate formed during an enzymatic reaction is. Transition structure is a combination of what? In any enzymatic reaction, you have the substrate and product. and in between you will have a substrate enzyme complex, which will transform and become an enzyme product complex, and then the enzyme gets separated from the product and the product is released, enzyme becomes free to bind to the substrate again. So there is substrate plus enzyme combines together to form the substrate enzyme complex, enzyme product complex, enzyme plus product. Enzyme is free to bind the substrate again. There are two intermediate transition states. What are the two intermediate transition states? Substrate enzyme complex and enzyme product complex. These two are intermediate states. Are they low energy states or high energy states? The transition states are high energy states. Intermediate formed, they are formed during the transition from substrate to product. Why is it transient? Transient means what? Look at the options. Transition state structure of the substrate formed during enzymatic reaction is transient but stable, permanent but unstable, transient and unstable, permanent and stable. There are two parameters. Its stability and its permanency. Transient means short-lived, permanent means long-lived. Stability, stable is remains as it is for long, unstable is changes quickly. So transition state in terms of its permanency, is it long-lived or short-lived? Short-lived. So it is transient. Then in terms of its stability, is it stable or unstable? Unstable. That is why it changes quickly. That which is stable doesn't change. So it is transient and unstable. A phosphoglyceride is always made up of. See, there is phosphoglyceride. There are three things there. What are the three things? In a phosphoglyceride, you will find three chemicals. What are the three chemicals or three structures? Phospho is a reference to phosphate group. There is some phosphate group involved. Glyceride is a reference to what? Glycerol, and glyceride is a combination of glycerol and fatty acids. Glycerol and fatty acids. So a glyceride is generally it can be of three types. A glyceride can be of three types. What are the three types of glycerides that you can find? Monoglyceride, diglyceride, and triglyceride. When I say triglyceride, how many glycerol and how many fatty acids you will find? One glycerol, three fatty acids. So a glyceride is always a combination of glycerol and fatty acid. It is not mentioned how many fatty acids here. Phosphoglyceride means there is definitely a phosphate group. So look at the options. Phosphate group should be present. Then glyceride is glycerol plus fatty acid. Fatty acid can be saturated or unsaturated. There is no issue about the glyceride should have only saturated or unsaturated fatty acids. It can be either of that. So glycerol is necessary, phosphate is necessary, fatty acid is necessary. Fatty acid can be saturated or unsaturated. These are the conditions we arrive at when we look at phosphoglyceride. Then look at the options now. Only a saturated fatty acid esterified to a glycerol molecule to which a phosphate group is also attached. Phosphate and glycerol is fine. But what is not acceptable in this statement? Only a saturated fatty acid. Not necessary, it can be unsaturated also. Option 2 says only an unsaturated fatty acid. Heterocysts, basal bodies, pneumatophores, chromatophores. See, just like that you look at it. Pigment is colored. Pigment is color. In all the four options, one thing you can definitely rule out. Pneumatophores. Pneumo means air. Something which contains air. So rule outs, even if you don't know what it means, you can rule out. Chromato refers to color. Force refers to compartments. Chromatophores are colored compartments. Fits well to our statement. Pigment containing membranous extensions in some cyanobacteria. Possible. You know basal bodies. What are basal bodies? Basal bodies are the structures from which flagella and cilia emerge. Flagella and cilia are attached to the basal body. 9+2 structure of cytoskeleton. So basal body is like the motor for flagella and cilia. So that is not the pigment containing body. Heterocysts. Heterocysts are what? What is special about heterocysts? Huh? Nitrogen fixing bodies. Heterocysts are specialized nitrogen fixing cells. They are nitrogen fixing cells that you find among cyanobacteria, in the cyanobacteria. Okay. So, easy question there. A major site for the synthesis of lipids. Smooth endoplasmic reticulum. Endoplasmic reticulum has the role of protein synthesis. Which endoplasmic reticulum participates in protein synthesis? Rough endoplasmic reticulum participates in protein synthesis. Smooth endoplasmic reticulum participates in lipid synthesis. Cholesterol production, lipid synthesis. Smooth endoplasmic reticulum. Modification, packaging and delivery happens at Golgi. Golgi complex is responsible for modification, packaging and delivery. So vesicles in the cell are produced by what? Vesicles are produced by Golgi complex. Golgi complex produces vesicles. Any vesicle means pouch, packet. Vesicle is a packet, a temporary packet. Alright. Just recognition and trying to understand the origin of each of these terms will help you get closer to the answer. There are possibilities that you don't know the question or the options. So how do you arrive close to the option, close to the right choice? By root analysis of the roots of the words. Break it down. No term is meaningless. Every term indicates what it refers to. Got it? Complex formed by a pair of synapsed homologous chromosomes. Synapsed homologous chromosomes is called what? Equatorial plate, kinetochore, bivalent, axoneme. Bivalent is the junction of synapsed homologous chromosomes. Do non-homologous chromosomes come in synapse? Can non-homologous chromosomes synapse? No. Synapse or crossing over. Crossing over leads to synapse. Synapse is what? The junction of contact. Synapse is the junction of contact between chromosomes. What kind of chromosomes? Homologous chromosomes. What is similar in homologous chromosomes? Can I say homologous chromosomes have the same nucleotide sequence? Homologous chromosomes have the same nucleotide sequence. Homologous chromosomes have the same nucleotide sequence. True or false? True?
Yes, sir.
Sister chromatids have the same nucleotide sequence. True or false?
Yes.
Hmm?
True, sir.
True. Chromatids of homologous chromosomes are non-sister chromatids. True or false? True. So chromatids of homologous chromosomes are non-sister chromatids. Now these two are homologous chromosomes. Each one has two chromatids. These two are homologous chromosomes. So they are non-sister chromatids. These two are sister chromatids. These two are sister chromatids. Correct? Sister chromatids, sister chromatids. Now put all the three statements together. First one, what? Homologous chromosomes. So these two are homologous chromosomes now, each with a pair of sister chromatids. Homologous chromosomes have the same nucleotide sequence. Sister chromatids have the same nucleotide sequence. Non-sister chromatids of the homologous pair have the same nucleotide sequence. What is right? Sister chromatids have the same nucleotide sequence, not homologous chromosomes. Homologous chromosomes don't have the same nucleotide sequence. Then what is same? They have the same gene sequence. Homologous chromosomes have the same gene sequence. They have the same loci sequence. The locus is the same. You understand? The sequence of different loci is the same, not the nucleotide sequence. Remember this. Homologous chromosomes are similar chromosomes, not exact copies. Sister chromatids are exact copies of each other. Why? Sister chromatids are formed because of the duplication of DNA, and they remain together until they are separated. Sister chromatids are exact photocopies. They are exactly similar to each other. Homologous chromosomes are exactly similar in terms of the gene sequence, locus, sequence of different loci. Alright, that is why you can have different alleles on the two homologous chromosomes. Otherwise, the different alleles will not have the same nucleotide sequence. No, alleles will have variation in the nucleotide sequence, which give them the differentiation in terms of their expression. Understand? So equatorial plate is what? When do you see the equatorial plate being formed? Metaphase. In meiosis, in which stage of meiosis do you find? In meiosis 1 or meiosis 2 you find equatorial plate? Meiosis 1. What about in meiosis 2? Both. See, I am giving you an either or question. The answer need not be either or. It can be both or neither. Alright. So you get drawn into that. Both. Metaphase is in metaphase there is equatorial plate formation. What is kinetochore? Kinetochore.
The center of junction of spindle fiber which is present at the center.
At the centromere you find the okay, kinetochore is DNA. True or false? Kinetochore is DNA. False. Kinetochore is RNA. Kinetochore is protein. True. Kinetochore is protein. It is the cover. It is the proteinaceous protection around the centromere region, holding on to which the spindle fibers pull the chromosomes apart. What is axoneme? Axoneme. Core of the cilia or flagella. Good. Okay. Next one. 3 boxes in this diagram represent our favorite item. 3 major biosynthetic pathways in aerobic expression. Arrow number 4, 8 and 12 can all be. A is glycolysis, B is Krebs cycle, C is electron transport chain, is it? C cannot be electron transport chain if we have to make the right choice as NADP or NADH. Correct? See, A is glycolysis, B should be oxidative decarboxylation, C is Krebs cycle. C is Krebs cycle. So, because look at the things, NADH, ATP, water, FAD or FADH2 are the options. In if ABC were to be glycolysis, Krebs cycle and electron transport chain, 12 cannot be NADH or FADH2. Correct? 12 cannot be NADH or FADH2. Right? 12 has to be ATP and water. It has to be ATP or water. Glycolysis produces 2 ATP and 2 NADH. That is the final output. Krebs cycle also produces ATP. Does Krebs cycle produce ATP directly? Krebs cycle produce GTP. GTP. So B cannot be 8. If we are assuming B to be Krebs cycle, 8 cannot be ATP. Krebs cycle is not producing ATP. Glycolysis and Krebs cycle do not produce water as a byproduct. So we have to consider NADH or FADH2 as the right choice among all three. If C is electron transport chain, C cannot be, 12 cannot be NADH or FADH2 because NADH and FADH2 goes into the electron transport chain as electron donors. They are not coming out. Correct? So ABC cannot be glycolysis, Krebs cycle and electron transport chain. ABC has to be glycolysis, oxidative decarboxylation and Krebs cycle. Then 4, 8 and 12 can be NADH. Glycolysis does not produce FADH2. Neither does it produce water. So 3 and 4 ruled out. For A assumed to be glycolysis. NADH and ATP is produced. In B, pathway B, we are considering it to be oxidative decarboxylation. ATP is not produced, but NADH is produced. Pathway C considered as Krebs cycle, if we look at it, Krebs cycle produces NADH, FADH2 and GTP. So NADH is anyways there. By this logic, you have to conclude that 4, 8 and 12 are NADH. Understand? So ABC, the question can be reversed. If pathway A, B, C are the three major biosynthetic pathways in the glucose metabolism, three major biosynthetic pathways in aerobic respiration, and 4, 8 and 12 is NADH, what is ABC? That can be the question. That can be the question in different combinations. One of the combination will be glycolysis, Krebs cycle and electron transport chain, which most of the students will write. Because you know these are the three components of the aerobic respiration pathway, and NADH you know is associated with all three. You will not pay attention to NADH 12 coming out of pathway C, and you are more likely to select glycolysis, Krebs cycle and electron transport chain, and skip and the single step oxidative decarboxylation will skip the mind. That would have been the right choice, right question to be framed. But anyways, here, did you understand the logic, all of you? Are you understanding it? Okay. The most abundant intracellular cation is potassium, extracellular is sodium. Extracellular cation, see ions, potassium, sodium, calcium. These three are important. Cardiac activity, potassium. Nervous system, sodium. Sodium, potassium, both are important, influx and efflux. But sodium is more important for nervous system activity. Muscle activation and muscle contraction, calcium. Muscle contraction, calcium. Alright? Sodium also you have to remember with glucose absorption. Glucose absorption, remember that. Okay. Um, during resting membrane potential in a nerve fiber, intracellular cation will be which one? What will be more inside the cell? Potassium. Sodium will be more outside the cell. What will happen at the threshold potential? What will happen at the threshold potential during in a nerve cell? When the nerve cell attains threshold potential, what will happen? What ion movement happens and in which direction? You tell me that first. What ion moves and in which direction? Sodium moves from outside to inside. Who allows this to happen? Voltage gated sodium channel. All right, okay. We'll not go more into that, we'll see it later. During seed germination, its stored food is mobilized by who mobilizes stored food from seeds? Mobilizing stored food from seeds is a part of breaking the seed dormancy. Who breaks seed dormancy? Gibberellin. Gibberellin breaks seed dormancy. So, stored food mobilization and contributing to initiation of germination, gibberellin. What is the role of ethylene? Fruit ripening. Ethylene is responsible for fruit ripening. Cytokinins? Kinin is to accelerate or increase cell, increase cell division, breakdown, division of cell. Cytokinin will promote cell division and cell growth. Abscisic acid induces dormancy and inhibits growth. So seed did not germinate so far because of abscisic acid. Seed started germinating because of washout of abscisic acid and activation of gibberellin. Which of the following criteria does not pertain to facilitated transport? Requirement of special membrane proteins. Is it required? Facilitated transport is active or passive? Facilitated transport is passive. It is also called facilitated diffusion. Facilitated diffusion. It is passive transport. Do you need special membrane proteins for that? Yes. Facilitated transport happens with the help of special membrane proteins like glucose transporters in the intestine. Glucose transporter is a co-transporter. Glucose and sodium co-transporters. In the presence of sodium, glucose transportation is facilitated. Is it selective or generic? Facilitated transport. Selective, does not allow everybody. It is like VIP pass. If you have VIP pass, you can enter. You don't have to pay ticket cost also. All right. Selective. Transport saturation. Will there be saturation for facilitated transport? Will there be saturation? What do you mean by transport saturation? See, simple. Some specialized proteins are required for transporting a particular item. If the proteins are all engaged in association with that particular item to be transported, increasing the concentration of the material to be transported will not increase the transport because the mechanism is already saturated. Transport saturation happens. Uphill transport. Can facilitated transport happen uphill? Uphill means what? Uphill is against concentration gradient. Uphill transport is transportation against concentration gradient. That does not happen in facilitated transport. It is along concentration gradient. Okay. First stable product of fixation of atmospheric nitrogen in leguminous plant is nitrous oxide, ammonia, nitric oxide, glutamate. Enzyme nitrogenase fixes the nitrogen into ammonia, which is the first stable compound. Then ammonia is pushed into glutamate. It becomes a part of glutamate through assimilation. Then nitrate and nitrites are formed in the soil. They are absorbed by plants after the reaction of glutamate with some other compounds and becomes nitrate and nitric compounds. Ammonia is the first stable product. Which of the metabolites is common to the respiration mediated breakdown of fats, carbohydrates and proteins? Glucose 6-phosphate, fructose 1,6-bisphosphate, pyruvic acid, acetyl-CoA. What is common? Acetyl-CoA. Acetyl-CoA is the central molecule. It can go into carbohydrate synthesis, lipid synthesis or protein synthesis. Alright. Where does acetyl-CoA enter? For energy production, acetyl-CoA enters into Krebs cycle. Which one of the following statements is correct? Hard outer layer of pollen is called intine. Outer layer is exine. Sporogenous tissue is haploid. Sporogenous tissue is diploid. Endothecium produces the microspores. Endothecium will grow and bulge and burst open and release and cause the dehiscence of anther. It will lead to the dehiscence of anther, distribution of pollen grain, but not for maturation of or the production of microspores. Tapetum nourishes the developing pollen. True. Ooh. Okay. Okay. True. The product of sexual reproduction generally generates longer viability of seeds. Is viability linked to sexual reproduction? Not necessarily. See, the viability decreases with repeated inbreeding. With repeated inbreeding, what do you call that? The vigor, hybrid vigor decreases. The vigor goes down, but viability is not dependent on sexual reproduction. Prolonged dormancy, new genetic combinations leading to variation, large biomass. Genetic variation. Sexual reproduction brings about genetic variations. Variations, what kind of variations happen will determine what kind of characters are expressed by the plant or its products. Sexual reproduction as such will not contribute to increased biomass or anything. Now you find this guava is this big. Each fruit will be some half kg. Alright. Big guava. Big in size, low on taste. More seeds, hard. We used to have one guava tree in our backyard. It used to produce only small, very small fruit. That's all, this much. No seeds, super tasty. You will just eat it like some small chocolate. Now this big guava, useless. Spineless pineapple. Spineless pineapple. Have you seen that? It is like a plastic cover around it. It looks like there are spines, but if you touch it, it's not spiny. The hybrid pineapple. They call it Maharaja pineapple. Anyways. Okay. Till 115 we have discussed. Rest of the questions we will discuss tomorrow in the morning session. Sleep well. Don't lose sleep. No continuing studies in the room. Sleep and wake up at 5 o'clock.